Gene knockout. Definition.
Gene knockout is a genetic technique that enables us to make a gene inoperative in an organism. This gene will lose its function by either avoiding the messenger RNA synthesis or shortening the protein sequence. These genes are called knockout genes, and the organism that contains it are called knockout organisms. The importance of this technique is that knocking out only affects the gene inactivated, while the rest of the genes are expressed normally because the technique is very specific.
Types.
We can define two types of gene knockout depending on number of inactivated genes. It can be a simple, double, triple, or quadruple knockouts, affecting from 1 up to 4 genes respectively. Number of alleles that the organism has. In haploids, gene knockout is more simple in comparison to diploid organisms that need several knockouts or breeding with homozygote organisms.
History.
In 1989, in the universities of Cardiff, North California, and Utah, these pioneering scientists started to research in gene targeting to create a knockout organism. The organism selected was a mouse that was created by the modification of embryonic stem cells. Later, the cells modified successfully were introduced inside the uterus of the mother. The newborns will show the effects caused by the gene deleted in vivo, observing the difference with the normal behavior. The contribution to the science was rewarded in 2009 with the Nobel Prize in Medicine and Laster Basic Medical Research Award.
Methods.
In the beginning, gene knockout was carried out by transposon mutagenesis. Then, other genetic engineering techniques have been used, such as bacterial artificial chromosomes, which are modified in vitro and then moved to an organism of interest. The problem is that these techniques were not very efficient, and that is why recombination has been one of the most important tools for producing gene knockouts.
Recombination is usually carried out by bacteriophage. Recombination is useful for deletions, point mutations, duplications, and inversions. Homologous recombination is the typical method for gene knockout. This method consists of interchanging a gene with a mutation and a drug resistance. Recombination can be done by using the protein CRE and LOXP sites. Gene knockout can also be done by site-specific nucleases. There are 3 methods: thin fingers, TALENs, and CRISPR. These 3 methods break DNA. Then, the repair mechanism of cells try to fix it by non-homologous end joining NHEJ to ligate 2 open ends. The repair mechanism, when it finishes imperfectly, it generates an insertion or deletion which results in a mutation. This mutation produces a non-functional protein, and it generates a knockout for the gene of interest. Thin finger nucleases are restriction enzymes very used in genetic engineering for creating breaks in the double strand. They act as a site-specific endonuclease that can bind to DNA at a specific site. Transcription activator-like effector nucleases, also called TALENs, are artificial restriction enzymes that consist of a DNA binding domain and a nuclease domain, FOC1. In order for the nuclease domain to cut, two different talents must be supplied to the cell, one targeting each strand of the DNA. When FOC1 cuts, it will cause a DNA break, which will be repaired by NHEJ, and will sometimes result in mutations that lead to a gene knockout. CRISPR is composed of a single guide RNA and a Cas9 nuclease. They come together to form a ribonucleoprotein, RNP. Cas9 recognizes a sequence in the genome, which differs depending on which species. After binding the DNA, Cas9 will cause a double-strand break. Then, NHEJ repair system will induce the mutations.
Uses.
The main uses of gene knockouts are studying genes' function by comparing the knockout organism to a wild type with similar genetic background so we can identify the function and contribution of the gene. Study drug or toxins degradation processes. Knockout organisms are also used as screening tools in developments of drugs to target specific biological processes or deficiencies. Alimentary industry. It could help improve transgenic agriculture and cattle. Medicine. Gene knockout has been tested in animals and it can correct damaged genes in humans and develop techniques to help cure cancer.
Concerns.
Many knockdown mice die while they're still embryos before the researcher has a chance to use the model for experimentation. The gene may serve a different function in adults other than developing embryos.
Some organizations have objections to the use of embryonic stem cells, causing ethical arguments. It may fail to produce an observable change in a mouse or may even produce different characteristics from those observed in humans in which the same gene is inactivated. It is not an efficient method for plant modification because of the insufficient frequency or efficiency of homologous recombination. To solve that problem, we can use gene targeting with transposons and bacterial recombination systems or through stimulation of the cell's own recombination processes.
Similar techniques.
Similar techniques of gene knockout are gene knockdown. The gene expression is reduced to minimum levels by the block introduction of most messenger RNA with interference RNA or by changing the promoter or regulatory protein. Gene knock-in. This technique involves a substitution of DNA sequence information in a genetic locus with the insertion of sequence information that is not in the locus.
Group members.
María, Miguel, Kiko, Mónica, Carmen.